So I'll get started now without wasting a lot of time. So the topic for the day is the role of big data in healthcare and how it has become a key to the personalized medicine. Right. So as Alok has already introduced, I am Dr. Jaspreet Kaur Dhanjal. I am assistant professor in the Department of Computational Biology at Indraprastha Institute of Information Technology, which is known as IIIT Delhi. So it's a very nice campus in the Oakland Industrial Estate Phase 3, New Delhi. And here I head a lab called the Translational Biology Lab. And the focus of our lab is in health informatics. And we work at the interface of computational and molecular biology. And if I broadly divide my work into two major domains, I would say I work in personalized medicine, and the other domain is drug design and discovery. So, in the personalized medicine, I am trying to understand disease pathologies in context of individual patients so that we can design some effective therapeutic interventions. So, I'll give you certain examples which make it more clear. But in very simple words, the same diseases that we already know, but we are trying to look at it with a fresh perspective. We are trying to see whether we can differentiate which of the patients can benefit more from our therapies and which of the patients require a different, an alternative therapeutic approach that can work the best for them, right? And in the second broad area of my work, I am interested in like finding out the proof of concept for the therapeutic potential of traditional medicinal compounds using modern science. So we all know we have a decade long history of traditional medicinal system. For India, if we talk about India, we talk about Ayurveda, right? So we know that Ayurveda has a very long recorded history. It is quite effective, but still it is not accepted at a global level because we do not know the way in which these herbal formulations actually work inside the body. So my aim over here is to look into the literature, find out clues, what are the therapeutic potential associated with different medicinal plants, and then using modern science, if I can prove the scientific basis for their activities, is one of my areas of interest. But today, for the today's talk, I'll be mainly focusing on the first part, that is personalized medicine, right? So a very basic slide to start with. We all know about this, right? So if I'm talking about living organisms, we are all made up of cells, which are the fundamental unit of life, right? And as humans, we have each of our cell, it has a DNA, which is the blueprint of how a cell will behave, right? So we all know about this. So DNA, it has a lot of instructions for a cell, how it has to grow, how it has to sustain itself, how it has to reproduce, and at what time it has to decide that it should go, it should get into cell death, right? So all these instructions are written in the DNA. And for writing all these instructions, especially for humans, we have a DNA which consists of 3.3 billion nucleotides, right? So 3.3 billion nucleotides or base pairs make our entire DNA, which has a lot of instructions. And we know that these instructions are decoded in the form of different RNAs and proteins. And finally, these proteins are the major players, right? So we have already, delineated the function of these different RNAs and proteins to a very large extent. And because of this information, we have been able to find out that what are the cause of different diseases, right? So for example, if I talk about the DNA in humans, right, 3.3 billion nucleotides, so many instructions already written there. But even a single nucleotide change can lead to life-threatening disorders, right? So this is just a schematic, a DNA with just one SNP shown over here. And in this case, it causes a life-threatening disorder, sickle cell anemia, right? One nucleotide change in the DNA which codes the instructions for making the beta globulin gene, right? So, but there are other disorders which do not have one change associated with them. A very simple example to start with is cancer, right? So we can say cancer is a disease of mutated genes. The outcome is the same. We are starting from normal cells. When we talk about cancer cells, we know that they have the ability to divide uncontrollably. That's the final outcome that we see. But when we look at different types of cancers, right, they are not actually the same. So how this cancer originates? We say the normal cell, it experiences some kind of stress, which leads to some damage in the DNA. So there is a small mutation. So say one nucleotide changes from the pool of 3.3 billion nucleotides, right? Now this mutation now leads to more mutations, right? Like mutation is not leading to more mutations, the first mutation, but then again, the damage is still there. The stress that the normal cell, that this transformed cell is experiencing still exists. And comes the second mutation. And if the stress persists for longer, then these mutations keep on accumulating. And finally, the cell, which gets the ability to, or gains the ability to divide uncontrollably, they survive and they actually cause cancer, right? So if you look at different cancers, the way in which these mutations have been accumulated is different, right? So it very clearly states that if you want to control cancer, we will have to look into the actual cause of the cancer, and we have to use therapy accordingly, right? So this is the very basic example that I can use to explain personalized medicine, right? So if I'm looking at multiple patients, I will have to look into the cause of mutations which led to this transformed, phenotype, uncontrolled division, and then accordingly, I will have to decide on which therapy to be given to the patients, right? But is it the only way of looking at personalized medicine? For example, I just talked about sickle cell anemia, right? There are many different disorders where the cause is just mutation in one gene, right? Or not even mutations. We know there is some protein which is getting affected, maybe not because of genetic factors, some other experimental factors, and it is leading to disease. So, do we have something that can be personalized in those cases as well, right? So, the answer is yes. Now, when I talk about any disorder, any disease, what we do as therapeutic, as a therapy, we try to design some therapeutic agents. They could be drugs, right? And when I talk about a drug, you administer this drug in the body, right? So upon administration of this drug, the first thing is the drug should be stable enough that it becomes available to the body, right? And then once it is available in the body, it is absorbed and distributed inside the cell, like in the entire body. And more importantly, that this drug, once it has entered into the body, it should not stay there for a very long duration. Like depending upon what is the purpose of the drug, the retention time could be different, but then it should not stay inside the body for a very long duration. Therefore comes the metabolism and elimination, right? So this is the first part of any drug, the effect of a drug on a body, right? And once it is able to stay there inside the body, it is absorbed, it is distributed, then comes what was the target for this drug, what is this drug actually supposed to do inside the cell, right? So there comes the binding to the molecular target. We call it the affinity of the drug for the molecular target. And then comes the efficacy, right? What is the mechanism of action? What dosage is required to make a desired change in the molecular target, right? So all these activities associated to drug can be divided into two different parts: pharmacokinetics and pharmacodynamics. The genomic signatures that I was talking about, right? They can be part of both the different, the streamlines. For example, say a disease, the cause is just one single mutation, right? But once a drug is being administered to cure that particular disease, there are certain genes which actually act at these levels. There are certain genes which help in making proteins which help in the absorption of this drug inside the cells. Then there are certain genes which help in eliminating these drugs from the body. So even if there is no genomic signature variation associated with the disease as such, there are certain other factors related to how the drugs that you are developing for curing this disease are actually absorbed and metabolized in the body, right? So there are multiple factors that can be taken into consideration, right? So up to this stage, we understand that there can be multiple causes for a particular disease. So you have to understand the cause of the disease and administer the therapy accordingly. Or the cause is just one, but the way in which the drug is acting in the body, that can vary depending upon variations in the other genes in the body, right? So it will become more clear once we move little forward. But if you have any questions in between, please feel free to unmute and ask the questions. I won't be able to see the chat, right? Okay. So, in nutshell, what is personalized medicine? I would say one size does not fit all, right? So if I have a cohort of patients, I have to analyze the needs of individual patients, and I'll have to administer the drug accordingly, right? So if say these are all cancer patients, some of the patients might respond to medicine A, right? Other group of patients or subset of patients might respond to medication B. There could be a small population that will respond to medicine C, but there would be some people who will not respond to any of the drugs, but they require some surgical interventions, right? So depending upon the personalized needs of the patients, we design therapies which will have some effective response in those patients, right? But for now, I have only talked about the influence of genomic signatures, right? Something in the DNA which is affecting the way in which the drug will respond or is actual cause of the disease, right? But apart from the genomic signatures, there can be other factors as well. For example, microbiota. There are a lot of drugs whose efficacy is affected by what microbiome is actually localized in your gut, right? So depending upon the colonies of the microorganisms in your gut, the the influence or the final outcome of the therapy gets affected. Then it could be the lifestyle or the diet you are following. Some drugs might be more effective when you are following a certain type of lifestyle, right? So all this also comes under personalized medicine. Or the environmental exposure, right? The drug that you are taking, it is affected by some of the other external factors that you are living in. So personalized medicine, a very, very broad term, and a lot of things come under this personalized medicine, right? But this field of science, personalized medicine, as such, this branch of study in healthcare, it's actually not new, right? I'll show you some of the classic examples. For example, this warfarin, right? So this drug we are using since 1954, right? So this is actually a blood thinning reagent. So like in some of the cases after the stroke, this drug is administered to reduce the chances of the secondary clots, right? So it is a blood thinning agent, therapeutic agent, right? Now when I talk about the action of warfarin, what this warfarin actually does in the body, it actually interacts with this protein, right? It is a vitamin K this is a vitamin K epoxide reductase, this protein. So warfarin actually interacts with this protein. And what this protein does, it just reduces vitamin K, and it is actually, it converts it into an active form that is involved in the blood clotting process. So if warfarin interacts with this, it inhibits the activity of it, and hence blood clotting process gets affected, right? So this is the actual target of warfarin, right? Now, the normal uh, gene, right? The normal gene that produces the wild type protein for this particular gene, if its expression is normal, the doses have been standardized and warfarin can be easily given. But when we talk about the same protein, and if there is a variation, right? If there is a mutation in this particular protein, the activity of this protein or the expression of this protein reduces to a lot extent, right? So because of the lower expression of the genes, fewer proteins are formed, and therefore the normal dose of warfarin cannot be given. And in these cases, we require lower doses of warfarin to be administered. Otherwise, it can lead to life-threatening adverse effects, right? So this is the pharmacodynamic part, where your actual target for which the drug has been designed has certain variations, right? So I'm talking about genomic signatures over here. Again, genetic variations. The target itself has certain variations which affect the dosage of the drug that should be given. Then you have pharmacokinetics. Say you have a wild type protein only, right? The patient has a wild type protein only, but when we administer this drug, right, it is in the form of two stereoisomers, R and S. And as I said, the drug cannot retain in the body for a very long time. It has to be eliminated also. And we have mechanisms, or we have certain genes in the body which help in doing this process, right? So this is one of the genes which help in the metabolism of this drug so that it can be eliminated, right? So this is the actually gene which makes a protein which helps in the elimination. So if this normal gene is present, then you can administer the normal doses which have been characterized, right? But if there are certain variations in this protein, then the drug cannot be metabolized. So that active form of drug will stay in the body for a long duration of time, for unintended duration of time, right? So if this variation is present in the reference genome or in the genome of the patient, in that case, we have to reduce the dosage of warfarin because its metabolism is getting reduced, right? And if you do not do this, there will be increased risk of bleeding, and definitely you have to reduce the dose of warfarin that needs to be given. So this is one of the drug for which you have to do the genotyping before you can give the drug. So you have to check for variations in this enzyme that leads to metabolism of the drug, and also the actual target of warfarin, right? So this is a very classic example of personalized medicine that uses warfarin, right? Now if I
Yes.
come to
Yeah.
the, yes.
Just a minute, ma'am. Ma'am, some students are raising hands. So, shall we
That's
take
what
the
I said,
question
right?
between or at
Because
the end?
I am in slideshow mode. I cannot look at the you know questions that they are asking. So please
Okay.
unmute yourself and ask. I don't have any problem in answering
Sure.
the questions in between.
Sure.
Yeah.
Yeah.
Ma'am,
Mhm.
how do we know that in a general population who is having a mutated type or a wild type?
So if you see a patient with certain symptoms where these kind of drugs are to be given, their genetic testing has to be done. So it's like not obvious. If a patient comes, you cannot directly look into the genotype. But then we have certain kits available where you can do small screening for the presence of what type of variation do you have at that particular position in the genome.
Okay, ma'am. Thank you.
Right? Yeah. Any other question? Okay. If
No,
not, yeah. Any question?
no, ma'am.
Okay. In that case, I'll just move forward. Please unmute yourself and you can ask the question wherever you feel like. Okay. So this is the other example, uh, Primaquine. So this is one of the drugs. This is also available for the treatment since 1946, right? And this drug is being used to manage malaria in some of the areas where it is an endemic, right? And this was routinely being used, but then there was a doctor who observed that he actually gave this drug to some soldiers who were suffering from malaria. They got cured of malaria, but after some time he observed that they were being diagnosed with jaundice and anemia, right? All the soldiers who were given this drug, like some of those, they were uh, like they were being diagnosed with jaundice and they were having anemia. And later on, it was found that this drug can lead to hemolysis of RBCs in the patients which have mutations in another gene, G6PD, right? So if you have mutations in this specific gene, in that case, you have to look into again the genotype of the patients in which this drug has to be given. So in general, like this is an X-linked gene. So if both the genes are normal, then there will be like very less hemolysis of the RBCs. But and if you get into the heterozygote state, one of the alleles is normal and one is having some defect, then you see 30 to 80% of hemolysis. But in the patients in which both the genes are defective, there is like they become very deficient in RBCs. There's a lot of a severe hemolysis is observed, right? So all these patients, they actually develop acute hemolytic anemia, right? And after that, a lot of studies were conducted, and this theory was actually formulated. And if now this Primaquine drug has to be administered to the patients, we have to do genotyping for the presence of the alleles in this specific gene, right? So this is one of the genes which protect the RBCs from different types of stress. So if this is defective, even slight stress on RBCs can lead to bursting of the cell, right? So this is another example, a classical example where you have to look into the genotype of patients. So this is example of personalized medicine. You have to look into the genotype of each and individual patient before you can actually give this drug. right? Now, another example where we talk about cancer again, right? So if we talk about like CML, right? Chronic myelogenous leukemia. So in this type of cancer, we have, like in normal cases, normal individuals, we have one gene on chromosome number 9. It's a proto-oncogene ABL, and then we have another region on chromosome number 22 known as BCR, right? But these chromosomes actually break, translocation happens, and this part, this ABL actually translocates to this chromosome number 22. Both these genes or both these locations come together and it starts expressing as a fusion protein. So two different regions of the chromosome come together and they start producing a fusion protein, right? So if I look at this protein, BCR-ABL, in general, it utilizes an ATP molecule, it gets activated, and then it interacts with its substrate and downstream signaling, not getting into the signaling pathway for now, right? But then we have a drug called imatinib, which can interact at this ATP binding site. And once it interacts, it stops the further signaling process, right? Now, this is one of the signatures for CML, this production of this fusion protein. But not all CML patients have this fusion protein, right? So imatinib is very useful for treating CML patients if at all they have this fusion protein. So this is also one of the drugs for which if it has to be administered, you have to do the genotyping to find out what is the like whether this fusion protein is present or absent in these patients, right? Again, an example of personalized medicine. But in all these three examples that I talked about, here the drug was developed first, it was given to the patients, and then it was seen that it is effective only in a subpopulation, right? And it gave us the importance of pharmacogenomics, right? How the genomic signatures have to be taken into consideration for effective therapies, right? But now we have to think the other way around. We don't want to invest in developing a lot of resources and then realizing they are working only in a subpopulation of in a sub category of patient population, right? So what we need is we want to analyze the genome of different patients from a set of diseases. We want to see what is the prevalence of different type of mutants and can we design therapies accordingly, right? Now we are using a different approach. Now, one of the important examples over here is cystic fibrosis, right? We all have heard of cystic fibrosis. So uh, this disease also happens because of variation in one gene, CFTR, right? Mutations happen in the CFTR. So this is a ion transporter, right? So it helps in maintaining like a proper concentration of salts and water and different surfaces of different organs, right? So if this is working fine, it allows the movement of salt and the homeostasis is maintained, right? But if it is mutated, it does not allow the passage of these salts and it leads to the formation of thick mucus in the membrane or like on the membrane of different organs, right? And this is also one of the life-threatening disorders. Now, when the patients for this specific disease, they were sequenced, the CFTR gene was sequenced, it was found that there are multiple mutations that can happen in this particular gene, leading to cystic fibrosis, right? And these different mutations, they have different effect on the function of the protein, right? So this chart summarizes that. So for example, if the mutation is present at 542 position, in that case, the mRNA is formed, but the protein itself is not synthesized, right? So in this class 1 patients, these mutations are leading to reduced synthesis of the protein itself, the transporter itself. We do not have enough transporters being expressed on the surface of the different membranes, right? Then there is another class where these mutations are prevalent, and here the protein is being synthesized, right? We know that the synthesis happens in the ER and then it has to be transported to the transmembrane, right? So from the ER, it has to be trafficked to a location where it will be performing its function. So in this class of patients, this trafficking does not happen. Then there is third class of patients where you have these mutations, protein is being formed, it is even trafficked, but these proteins have a problem. that for the transportation of these ions, there is a gate. There's a gate in this channel which closes and which opens and closes. So for these class of mutations, this closing and opening does not happen properly. So the gates, they tend to close all the time, right? Then again, another class where these mutations are more frequent. And here again, we have to, like, there is decreased conductance. Like, even though the transporters are open, but a very limited activity is happening across these channels. Similar to that, we have different types of classes. And again, somewhere you have, the correct splicing is not there. Protein is less, is being formed, which is less in quantity. Then the protein formed is not stable, or mRNA itself is not forming. So we have different classes of proteins, different classes of mutations. Each of these mutations is causing a different effect on the protein. Either it is not being synthesized. If it is being synthesized, it has some, problem in its proper functioning. So depending upon which class of mutations it is, can we help at least some subpopulation, right? So if the protein is not being formed, I cannot do a lot, right? But in this class of mutations, where the protein is being formed, but the protein is not able to keep itself open for the duration that is required. So we have come up, like, not we, but the scientific community has come up with the drug called ivacaftor, which binds to this protein. It helps to keep it open for a longer duration, right? So in this case of cystic fibrosis, we cannot help all the patients, but we can identify which patients have these specific mutations, and for them, we can administer this drug. Right? We might have drugs in the other categories also, but it is just to give you an idea that how, like, what are the different areas in which we can look at personalized medicine, right? Now, P53, I think we all have heard about this protein. This is one of the tumor suppressor proteins. If there are mutations in this protein, the cell loses its ability to control uncontrolled proliferation and can lead to cancer, right? So this P53 protein, it has a lot of functions. It can sense different types of stresses that a cell can experience, and accordingly, it generates a response, whether the cell should stop dividing, whether it has to undergo into angiogenesis, autophagy, cell cycle has to be arrested, the cells have to migrate to a new place, the cell should die, or the DNA has to be repaired, or there has to be alterations in the metabolism. So P53 coordinates a lot of activities. But this is one of the proteins which is most frequently mutated in different types of different types of cancers, right? But there are certain cancers where the P53 is not mutated, like it can be reactivated so that it can check some of its functions, right? Now, if I look into the mutational profile of P53 protein, more than 25,000 mutations are already known, right? And again, similar to CFTR, the different mutations have different effects on the protein. Now, I'm talking about only one class of mutations. There are certain mutations which just destabilize the structure of P53. P53 has to make a tetramer, and then it goes and interact with the DNA. So, and then it causes the transcription of genes which can do all those functions that I listed, right? So the protein itself is a little unstable, and it is unable to stay on the DNA for a longer duration. So there have been research where people have devised or designed small molecules which can interact with this unstable protein. It can stabilize it for a longer duration. And now this protein, mutated protein with small molecule attached to it, restores the ability, and it starts functioning like a wild type protein, right? Again, this becomes an example of personalized medicine. So if the cancer patient has mutation in P53, and if it belongs to this class of mutations, these kind of drugs can be administered, and then P53 can take certain measures to correct what are the problems in the DNA itself, right? Now, with all these examples, the one thing that we realized is the need for population scale data, right? If we have to characterize all the mutations, so if I'm talking about CFTR example, I need to sequence this gene from a lot of different patients, then analyze what are the different variations in these genes, and accordingly, I can do more research to find out what happens if this mutation is present, how the structure gets affected affected in case this mutation is present, right? So what I require is population scale big data, right? So for personalized medicine, big data becomes very, very important. So far, we have been doing a lot of research, we are collecting data, but with the recent advancements, the recent technology that we have, collecting data has become very easy, and we can actually collect a lot of complex data, and we have computational methods of analyzing this data, right? Not only computational method, a lot of experimental techniques are also used over there, right? Now, when I talk about big data in healthcare, so this is one of the statistics from a report from 2020, which says we used to collect 153 hexabyte of healthcare data in 2020 in 2013, which actually increased to 2,314 exabytes in 2020, right? And what is the source of this data? When we talk about this data, we have so many medical imaging data available to us, right? We have health medical records from different hospitals. Doctors used to use these records to communicate among themselves, right? They used to prepare charts with all the information related to the patients. Now, those records, those medical records have become a rich source of information because it has longitudinal data for a patient which was admitted in the hospital, right? Then a lot of medical devices which are placed onto the patient when the patient is in hospital, that collects a lot of data 24/7, right? Then we have wearables. We are wearing smartwatches. Most of the people today are wearing smartwatches. It collects so much of data for all the time you are wearing it, right? It records your pulse, your heart rate. Some of them even have sensors to record the conductance through your skin, right? The oxygen levels. All this information is being collected longitudinally. You are having time series data, right? Then all the pharmaceutical research, which they are looking at the response of patients to a given drug, right? So many studies are being done. Then search engine data, like what people are searching, what type of symptoms they are searching. If I'm talking about a diseased, area, then smartphones. Again, we have a lot of applications which collect the digital, data. Then insurance companies, they also keep records of the people who are insured in their company, right? They do a lot of tests, keep those records. Then what casualty these patients actually suffered, all that can be related. And the genome sequencing data, right? So this genome sequencing data will soon actually, it will be more than the data that we actually collect under astronomy. So we say astronomy is one of the studies which collects a lot of data, a lot of high resolution images. Now the amount of data that we are collecting from the genome sequencing projects will be even larger than that astronomy data, right? And then, as I said, genome sequencing, we are saying DNA is the blueprint. Everything that you can find in a cell's altered behavior of a cell, it can be found because of variations in the DNA. So a lot of population scale sequencing initiatives have been taken across the world. So you look at all these different regions from the world, they have initiated their own sequencing projects where they are trying to sequence their population, look into different variations that they find across population, variations which do not result into diseases, variations which result into diseases, right? And once this information is there, you can do a lot from this particular data. Not only that, India also started its Genome India project in 2020, right? And this is being led by multiple institutions in collaboration. You can see all these institutions or partners listed over here, very renowned institutes participating, and some of the objectives of this particular initiative. The first objective, very important objective, is to sequence 10,000 Indian population, right? Now, 10,000 Indian people as a population. And then this will help us to catalog all the genetic variations in Indians that can help us identify disease causing genes. So we say that if we are talking about mutations, they also like there are different frequencies of mutations depending upon the place from where the population has been sequenced, right? We see that ethnicity also affects the frequency of mutants that we generally see, right? And these are the two main basic objectives that, sorry, not only two, three. So all these genetic bases for adverse effects and response to drugs also. So that is also one of the important part of personalized medicine. like I already gave you example of warfarin, right? Primaquine. So the same drug, but the response from the patients is different depending upon the genetic basis, right? So these are the three major objectives which can we can directly relate to personalized research. Apart from that, definitely capacity building, training people who can handle genomic data and who can develop resources to analyze this data. And then again, how can we develop resources for cheaper genetic studies? So all this comes under the objectives of this Genome India project, right? So you can like imagine or you can estimate the power of the genomic data and why it is important to sequence the indigenous population, right? Now, I gave you a lot of examples just to set the background what personalized medicine is. Just quickly, I'll give you some more examples from the work which is going in my lab right now, right? So one of the major focuses of personalized medicine research in lab again is cancer therapeutics, right? So as I said, when we talk about cancer, we have a lot of like we have tremendous work done in this area. We have a lot of remarkable success stories. We have targeted therapies that have been developed, right? Even though we have done a lot of research, we have a lot of outcomes. When we talk about therapies, the current therapies, they do not have equal response in all the patients, right? So the conventional therapies that we have, they do not like perform the same in all the population. And why? I already gave you the reason behind it. It's the level of heterogeneity, right? So as I said, cancer is a disease of mutations. What mutations accumulate can lead to different type of cancer, right? So if I talk about different types of cancer, say this is colon cancer, say this is your breast cancer, so this is your liver cancer. So if I talk about different types of cancer, the mutations which have driven this transformation of normal cells to malignant cells, it's different, right? So this is a very high level, like a very initial level if I talk about. But then when you talk about one type of cancer and you look at patients within this type, they are also very different from each other, right? Then if you talk about a single patient, you have tumors from two different sites in the body from the same patient. Those two tumors are also different from each other. There'll be like different dominance of population of cells in two different tumors, right? So when we are talking about heterogeneity, cancer is very, very heterogeneous. Not even two tumors from the same patient have same type of variation, right? So how can we imagine that the same therapies will work the same for all the patients, right? Now, to look into this, we have different technologies available that can help us, utilize the advancements in the research that is going on. For example, we can talk about genomic data, transcriptomic data, proteomic data, metabolomic data, lipidomic data, imaging, right? So we have so many different advancing technologies. I will not get into the technicalities of these, but you can say that these help us to generate high throughput data from a very large population, right? So we are also utilizing these different types of data which are available in open source. And the specific problem that we are trying to address, I'll explain it with the help of example again. Now, when we talk about the cells, so we are under constant stress, right? So many different environmental stresses are there around, right? Our cells, they constantly get damaged, and the DNA, there are certain mutations that happen, but then the cell has the ability to correct those mutations also, so that it can actually look into like it can divide as original DNA, right? So this is one of the proteins called PAR, which helps in modifying the bases, which helps in repairing the modified bases in the DNA before it is replicated for further processes, right? Now, it was observed that if this gene is mutated, right, PARP is not functioning, there is another protein in the body called BRCA, which takes over the function. BRCA has its own function. It does homology directed repair. But in case PARP is not working, then this BRCA, it takes over the function of PARP and it can modify those bases so that the replication happens. When the replication happens, the DNA is correct, right? Then scientists actually realized this and they utilized it to design a certain therapy. They observed that BRCA is one of the most frequently mutated gene in breast and ovarian cancers, right? So they said if BRCA is already mutated, that means PARP is helping the cell to survive. What if they block PARP also? So if they block both the genes which are required for repairing the DNA damage repair, then there will be so many mutations accumulating in the cell that the cell will not be able to stabilize itself, and it will have to be directed into apoptosis, right? So this olaparib is one such drug that they use, right? So when we talk about cancer, a lot of essential genes also get mutated. But then cancer cell has to make sure that the other gene that is getting mutation should have, like, if one of the essential genes is mutated, then it should have some other gene functional that can take over the function and help it to survive, right? So this type of concept is known as synthetic lethality. We say if gene 1 is mutated, the cell survives. Gene 2 is mutated, the cell still survives. But if both the genes are mutated together, cell cannot survive and it goes to death, right? And we are utilizing the same concept. So if a cancer cell has to keep one of these two genes alive, right? So we can capture all these events in the form of mutually exclusive mutations. So if gene A is mutated, you will not find any of the cells where gene B is mutated. If gene B is mutated, you will not find any cell where gene A is mutated, right? So this becomes a problem where you can look into mutually exclusive events. So this is what we are trying to do. We are analyzing genomic data from a very large number of patients, right? We are analyzing the variations or the mutated genes in that, and we are trying to find out genes. So these are different patients, these are different mutations. So I'm saying if this gene is mutated in different patients, right? This gene, then you don't find any patient which has mutation in these genes, right? So if this gene is mutated, this will not be mutated, and if this is mutated, this is not mutated. So that means these are two mutually exclusive genes. And therefore, if I find any cancer patient which already has mutation in this gene, I can use a drug which can stop the function of this gene also, and therefore the cancer cell will be forced to apoptosis, forced into the process of apoptosis. So this is one of the projects that we are doing right now, and we have found some leads. For example, Carmel2 is a gene which is not associated with cancer so far, but we know that it helps in cell migration in other normal physiological processes. And we found that it is mutually exclusive, like it is mutated in mutually exclusive manner with KRAS, which is one of the most frequently mutated gene in cancer. So we will be taking it forward to study its, further importance in cancer cells, and whether targeting this is a useful like it will be useful therapy or not, right?
Ma'am, I
Yes.
would like to know that the orange cells, orange segments in this graph, they represent the non-viable cells.
No,
So
no,
which
no,
are
they
not
are
viable?
co-occurring mutations. So the example that I gave you, I only explained you the significance of mutually exclusive mutations. But not only mutually exclusive mutations, we can also look at co-occurring mutations. So for
They
example,
have
if there is,
LOF and GOF both. The
yes,
orange
we can
ones, they
have
have
both.
the orange one, they have LOF and GOF, both
Both,
of
yes.
these.
Yes. So if I have to clarify this, I can give you one example. Say I'm saying there are two genes which are mutated, always mutated together. But I see that there is one gene, there are only deletions, and the other gene which has mutation is always amplification, right? So if one gene, there are mutations which lead to loss of function, there is the other gene where there are mutations for gain of function. So probably if this is deleted, I require the other gene to be amplified so that it can do its own function as well as the function of that deleted gene. So that becomes an example where I can say that co-occurring mutations are also important.
Okay, so which cells are represented the dead cells? Which color represents the
No,
dead
no,
cells
these are
here?
not dead. These are not cell data at all. So these are simply
Okay.
gene and mutations. So I'm saying, say this is my gene one. So I'm saying black color means it is mutated. Gene one is mutated in 3 patients, right? Then, like the first patient, I would say. Then I can say this gene one is not mutated in this patient. It is not mutated. So black denotes mutation, gray
Mm.
denotes not mutated, right? And actually this was also black, but to find out patterns, I'm just showing you patterns. So if this gene was mutated in these number of patients, you don't find this gene to be mutated in these patients. So that means this might be an important gene in the absence of this gene.
Okay.
Does this make sense? So if this is PAR, the example that I gave you, if PARP is mutated, BRCA has to be functional. And if your BRCA is mutated, then this PARP has to be functional. So in these set of patients, this PARP is mutated, but this BRCA, it is functional. And in these set of patients, this BRCA is mutated and this PARP is functional.
Okay.
Right? So these are mutually exclusive patterns that we can find. So we know that one of these two genes has to be functional so that the cell can survive. So now if I know my patient has PAR mutation in PAR, I know that if I block BRCA also, the cell will not be able to survive. Right? But in normal
Yeah.
cells, the PARP is functional. So even if I block BRCA, the PARP can do the function. So normal cells will not get affected, but cancer cells selectively get killed. So this is
Okay.
the advantage of using synthetic lethality.
Okay. Thank you.
Right? Okay. So I'll rush a little. So the other research that we are doing right now, it is similar to the cystic fibrosis. That was the motivation. So there is another disease called Wilson's disease. Here also, the cause of the disease is mutation in one of the transporters. So this ATP7B protein, it helps in eliminating excess copper from the cell. So in some of the people, this protein does not work. Because of this, there is copper accumulation in the body, mainly in liver, brain, and eyes. So these are the initial symptoms. But if it is not diagnosed in time, it can lead to neurological disorders. In the brain also, the copper starts accumulating. So the only way of treating this disorder, once diagnosed, is using copper chelators. So we have small molecules that go bind with copper in the cells, and they make it in a form that they do not require ATP7B to excrete it out of the cells. And for that, we have only 3 FDA approved drugs available right now. And this is the most commonly used drug. If we in India, if we have to use this drug and we have to manufacture it, the active ingredient required for this drug, we import it from China. And in 2016, there was a ban on import of the material from China. And at that point of time, these Wilson patients, they suffered a lot. There was a shortage of this drug. And for these patients, lifelong, they have to rely on these copper chelators for, eliminating the copper from their body. But if they keep on taking this medicine, they will be fine. But the treatment regime is very hard, right? The time duration for this they have to take this medicine is very hard. So we wanted to look into what are the other ways in which we can help. So one of the ways is look for more different types of copper chelators that can be produced in India, the same treatment regime that is possible. But then we wanted to see whether can we help even if a small population, can we help correcting the protein itself? We don't have to give copper chelators. If the protein starts functioning properly, then the patients don't have to worry. Normal physiological function will be activated. So similar to CFTR, there are more than 500 different mutations already characterized for ATP7B. So what we are trying to do is we are trying to develop a computational model which can mimic, right? We are just making a computational model of it. We are trying to simulate it in the presence of copper, and we want to see how the wild type protein actually functions inside the cells. And then we plan to introduce these mutations, 500 mutations one by one, and then simulate the protein again and see what happens to the protein. Why is it not able to take out the copper? And then can we find that small class of protein mutations which locally distort the structure? For example, similar to P53, right? The protein itself is unstable. If we provide little help from outside, the protein will become stable. Or that drug for CFTR. If there is anything that is not letting the copper go out of the protein, and if we can make a small molecule drug that can help in restoring the normal physiological function of the protein. So this is also one of the projects going inside the lab, right? So not getting into more details, we are almost towards the completion of it. So all the examples that I talked about till now, they all were related to the therapeutics, right? The drugs that we are giving. Either you have to look into the genomic signature so that you know whether the drug will be metabolized properly or not, it can be eliminated or not, there are no side effects associated with it, or you are actually looking into the genomic signatures to see whether this drug will actually go and bind to the target or not, right? But personalized medicine is not only for therapeutics. There are many different ways in which you can help the patients. For example, the diagnosis itself. You can have personalized diagnosis for the different patients, or you can have prevention. You can have personalized ways in which you can prevent a disease for a patient, right? So I could not have covered, like, this is a very vast topic. I could not have covered more. So I'm just giving you some more hints. For example, as I said, not only therapeutics, it can also be used for better diagnosis. So this is one of the very interesting studies where one of the doctors, he has followed his ovarian cancer patients for a longitudinal study. And this is one of the proteins, cancer antigen 125. So this is the way we have, different enzymes being estimated for diagnosing liver disorders and all. So for ovarian cancer, this is one of the protein which is estimated, right? And the normal threshold is 35 units per ml. So this is generally used to see if the levels are elevated, there are chances that the patient might be suffering from cancer. So he has followed a lot of ovarian cancer patients, and he has actually shown that one of the patients, for that patient, this level was never reached. This CA 125 level was always less than the threshold which has been decided based on population studies. And there was a lot of fluctuation in the CA 125 levels of all the different patients. So overall, he concluded that we cannot rely on these population thresholds, though they are a good measure, but still it has to be personalized for individual patients. So you can just have a look at that paper, very simple, but it gives you the importance of personalized thresholds also, right? Similar to that, people are now using data from variable sensors, as I said, smartwatch, right? So we generally say, like if we are talking about the blood, like blood pressure, right? We monitor the blood pressure, and I say systolic blood pressure greater than 140, it's the indicator of hypertension, right? But for some people, it's normally, like for normal also, their BP is generally higher than this. And smartwatches actually capture this data longitudinally for us. So can we use the data from smartwatches, create certain models which can create what is my personalized, threshold? If my BP goes above this, then I have hypertension. Maybe for some people, the blood pressure is always lower than that. So even slight increase can be indication of hypertension, right? So a lot of study is going around in this area. Then, as I said, how will a drug respond to a given patient, right? What are the genomic signatures which affect the response to any drug? A lot of different studies are there. So this is one of the papers on the basis of experimental techniques. Then this is another paper which makes use of artificial intelligence to do the same. You have drug response, you have genomic profiles, a lot of patients, and you are using machine learning, artificial intelligence to actually predict what will be the response if given a new patient's data, right? So personalized medicine has a lot of scope, lot of different domains in which it can help you. This was just a very little insight, just to give you an idea of what all it is about. And I think this will help you to get into like this branch little bit in more detail, and then you can follow up on the recent advancements that are being done in this field, right? So to summarize, what are the benefits of personalized medicine? First of all, the effective medication, because you are tailoring the treatment to the characteristics of each and every patient. So therefore, they would respond to the given treatment in a much better way, right? It reduces side effects, right? For example, if you're giving warfarin to a patient without looking at their genomic profile, there are higher risks of increased bleeding and the patient might die, right? So we reduce the adverse event risks to a large extent. Then it lowers the healthcare cost. You are using the therapies which are effective for the patient, so you don't have to go through a lot of hit and trial. You don't have to give a lot of drugs to the patient. Okay, this is not working. Let's move on to the next medication. So the time for which they are hospitalized, it reduces, and also the medicine cost, right? Early diagnosis, early prevention. One of the benefits, for example, I talked about CA 125, right? If we know what is the personalized baseline for me, right, then a little fluctuation from that can, help me indicate if at all there are any unfortunate circumstances, Then improve disease management with the help of variable sensors or mobile health applications. I can do for a lot of different, regular tests. I can actually look into the personalized thresholds based on my variable sensors data. One of the most important applications is designing clinical trials. Many of the drugs fail because they are useful only for a subpopulation, and I do not know. For example, say I was doing clinical trial for this imatinib, right? And I have a lot of CML patients in my clinical trial. I'm giving this drug to all these patients and seeing the outcome. But from all the CML patients that I had in my clinical trial, say only 5 people had this fusion protein. So this drug will work only for 5 patients and it will be declared ineffective, right? So if I already know what are the limitations of my drug, I can choose the people in the clinical trial accordingly, the patients accordingly, and the trial will go successfully with better chances of the drug getting accepted, right? So these are some of the limited applications of personalized medicine. Definitely the scope is much beyond the vision that we have right now, right? And I'm sure you all will help us to take this into more advanced stages. With this, we come to an end. Thank you so much. And I thank you all for your patience, listening. And I thank the organizers for giving me this opportunity to share this knowledge with you all. And definitely IIITD, my colleagues and my team of students who are helping me to, carry out research in the domain of personalized medicine.